Hello everyone, I am Santosh from IIT Kanpur. Before I start, I would like to thank the organizers for giving me this opportunity to interact such a knowledgeable audience. A special thanks to Professor Chaturvedi. So now, without further ado, uh, today's topic, which I chose, it's kind of introducing a new kind of material which is finally going to be decided by people like you that whether it qualifies to be a material for biological interest or not. We as researchers in different technology institutes, different scientific research institutes, we have our own hypotheses, on our ideas. We try to develop different things. Many a times it's the fascination of that material which pulls us in a particular direction. But the validation comes from the people like you who actually take it to the next level, who take it to the site where it is going to be used. And if we get suggestion, criticism, comments on those presentations, then we feel like, okay, now we are engaging with the right kind of people. So I would be expecting all this from this August audience. So let's start. I am associated with 3 different centers. I am faculty at Biological Sciences Bioengineering Department. Along with it, there are 2 more centers which have recently come up in IIT Kanpur. One is Gangawali School of Medical Sciences and Technology, which has the vision of bridging this gap between the medical centers and research labs. Another is Mehta Family Center for Engineering in Medicine, which is, you can say, extension of our bioengineering department. These are the people who actually work with us. So our current team has multiple PhD scholars, postdocs, RA, MTechs, and even BTech researchers who are doing their degree and they do contribute in direction of research. Our collaborators, very important people who actually control us when we are running behind something without any particular aim or without any particular control, who tell us that, okay, no, this is correct or this is not. Because without being multidisciplinary, you cannot be successful in any of the fields. Then our funders, very important agencies who support our ideas. Sometimes these are very crazy ideas. And if you don't believe me, ask to my PhD scholars. So why carbon-based system? So if we just think about the carbon, then first thing comes to our mind that, okay, if we start from our house, if we go for our old cooking style of chulha, then there is production of this carbon shoot. Then there is kajal, surma, different things. These all are carbon materials. Then we come out to go to our office or our labs, and then we are experiences the production of this carbon from automobile, a lot of carbon from factories and all. Then it just simply comes to our mind that, okay, it's something very negative. It cannot be something which can be utilized in biological system for some benefit. Now, this material has a lot of potential, but struggling with this kind of, you can say, given signature that, oh, this is something bad for biological system. Now, some of the researchers in last decade, in the beginning of last decade, took it as a challenge, and they thought, okay, let's check when whole body is made up of carbon as atom or element, and it is just threading with carbon, with hydrogen, oxygen, phosphorus, sulfur, and creating different molecules. How can carbon itself be that bad? there must be something in those carbon materials which could be used for the benefit of the biological systems and ultimate benefit of the human being, because we are the one of most greedy most species in this world. We always think about what is good for the human being. So that is the criteria that, okay, whether it is good for human system or not. Then they started coming up with different kind of molecules which are based on carbon, and carbon itself was found in multiple allotropic forms. So it was either carbon nanotube, it was either graphene, it was buckyballs, and all these materials. The biggest challenge these carbon material scientists they faced, that it was not soluble in water. And anything which is not soluble in water, either you have to assist the solubilization by using something which is organic, and that even if it has excipient, it will create toxicity, or you change the carbon material so that it becomes soluble in water. Then different strategies came, and they started generating a new form of carbon material which is called carbon nanoparticles. Now these carbon nanoparticles, luckily, they are suspendable in water system. So you can suspend it in regular water, you can suspend in buffers of different pH, you can suspend in different growth mediums and different body fluids. Now that was the barrier which broke the restriction of carbon material in non-biological system use, and it came to biological system. Now today I'm going to talk about few of the properties of these carbon nanoparticles and see whether it is qualifying to be a new hope for the biological bio-nano-medicine for use in clinics. Here there are some examples quoted here. So they can be, this is just the highlight so that we can get more engaged in this material. So it can be used for bioimaging, it can be used for biosensing, it can be used for drug delivery, it can be used for making smart polymeric materials, gels, transdermal therapeutic, and all that. Now, if it is so promising, how easy or difficult is the preparation of this material? Because that will be the first bottleneck which one want to handle. Because if it is very difficult to produce, then its cost is going to be higher, and then it will be less available, more skill set would be required to produce it, and so on. Luckily, it is very easy to produce. If you have a hot plate in your lab, you can produce it. Forget about hot plate. If you have a kitchen microwave in your lab, you can produce it. Even forget about that. If I say that every day, if you are baking something in your kitchen, you are actually producing these carbon nanoparticles. So I'm not saying wrong. Because whenever you burn any sugar or any carbohydrate or any protein or any fat, ultimately you are producing carbon nanoparticles. Now the only trick is that how to control the synthesis, make it reproducible and isolate it, because there will be a lot of chunk of carbon which will be produced, and that you cannot use as in any of these applications, for any of these applications. Now, if you want to really make it more sophisticated, more controlled, then there are microwave synthesizers which can be used. So generally in our lab condition, we use it. But sometimes for experimental purposes, for scaling up the process, we also use the hot plate method or kitchen microwave method, so that we can see how easily it can be produced. Now there could be a question that, okay, if you are using very purified carbohydrate material, it could be very costly to produce. So there is alternative. There are multiple byproducts of different sugar industries and carbohydrate industries and protein industries, food industries, which can be used as raw material to produce it, like molasses, like syrup, like nectar, like honey, like peel of banana, even peel of different fruits. They can be used to generate these carbon materials. Only thing is, yield percentage, quality of particle, size, their surface chemistry, that might vary. That you have to tune according to your requirement. Now, if they have to qualify to be good for biological system, they need to interact with cellular system. Now, do they interact? So answer is yes, they do interact. They enter cellular compartment and generally they take endocytotic pathway. So that has been proven in one of our studies. We could prove it. We use different kind of blockers of different entry routes, and we saw that okay, which are the particular pathways it is following. Now it depended on that what kind of surface chemistry you are generating during the preparation. So that tells that there is a tunability. So it's not that whatever the original material you are using to produce them, it's also that after producing you can coat with different kind of materials and then tune the property. And that will be entering in the cell by different pathways. Then you want to know that how you can use it for bioimaging application. So these particles, these carbon materials, these carbon nanoparticles, they are autofluorescent. So what they do, if you excite them in the range of 350 to 550, they start emitting light in just plus 50 nanometer of wavelength. So you start 350, you get light at 400. You excite 400, you get light at 450. You excite 550, you get light 600. Now that is the limit where one need to push it forward, because we know that for our biological tissue, our blood, the maximum absorption of light in that range 500 to 600. So you want to push it beyond. For that you have to incorporate or passivate different kind of molecules, polymers, dye molecules, pigments, so that it can be shifted toward near IR, and then it becomes a very magical bioimaging agent, which can be also used for 2-photon imaging, which can be used for deep tissue penetrated system and imaging. Now, how would you know and how would you utilize it in your cellular system? So there is an on-off system can be done, because many of the physicists, they did multiple research and found out that whatever fluorescence or luminescence is coming out of this system is surface phenomenon. And if you decorate the surface with any molecule which can quench those scatters created on the surface, then it will be off. And then as soon it goes away, those molecules are going away, it switches on. So if you want to follow any biological phenomenon inside the cell, where there is a particular molecule which can make association with your carbon nanoparticles, then it can work as a switch on and switch off based on the which stage of that biological phenomenon you are in. Okay, so that's how it can work. We could investigate it and publish it. Then how to change the surface passage? It's very easy. You can do during the preparation of carbon particles or after the preparation of particles. So suppose you are interested in a large molecule or large protein or large gene sequence which you want to send inside the biological system. You prepare these particles and then decorate those polymeric materials, could be biopolymer or synthetic polymer, and then that wrapped system, you send it inside the cell. Now when it will reach to endosomal cavity, there because of the presence of these anionic lipids, it starts interaction, and then because of ion pairing, during the ion pairing process, it kicks out those molecules which are wrapped around. Now, if it is a protein system, then you do that kind, you use it for that particular therapy process. If it is gene sequence, then it can go either you might be delivering siRNA, shRNA, or you want to deliver the DNA, which can go inside the nucleus and start the process. So it is very easy. Now, if you are doing co-preparation, you can even tune the size. The size could vary from 2 nanometer to 200 nanometer. Now that also is very reproducible and yield is also very nice. So it depends on whether you want to use it for just biofluid purposes, or you want to deliver some content inside the cell, you can decide, and based on the pre-decided composition you can get a size, right? So in generally nanomedicine, the concept is if you want to deliver something inside the cell, then the size should be close to 100 or less than 100, because the leaky vasculature, like in case of the cancer, right, if you want to deliver a drug loaded on these nanoparticles, it has to cross that leaky vasculature, it should be less than 100. But in some other cases, even the larger one would work when you do not, where you don't want to go inside the cell, right? So size variation can be done. Now, what about if you want to use intrinsic property of some medicinal plants and you don't want to extract that medicine or that active component from the plant, but still you want it to be incorporated in the nanosystem? You use that raw plant part. It could be leaf, it could be crust stem, it could be roots, it could be any part of the plant or a plant as such, and then convert it to carbon nanoparticle. You'll find that many of those particles, they will inculcate that medicinal property. Now there could be a question that what about the heat you are generating or the microwave you are exposing it to, that might degrade the medicinal value of that particular active component. So this is a very genuine concern. So the option is that you do microwave preparation. Many of these thermolabile materials, they are stable in microwave. So that increases the bandwidth of this process. And then more and more materials and more active compounds can survive. So here we used an algae for preparing this carbon particle, and these carbon particles were so good by themselves, they had the property of controlling the migration of the cells. So if there is a chance of metastasis, then if this particle itself is used directly without adding any drug, it works as anti-migratory. Now, yeah, how they do interact with cellular membrane? Now, by themselves, they will not stay in the membrane for a long time. But if you want it to stay there, you just conjugate a long chain fatty acid on the surface. And then because of the fatty acid chain, hydrocarbon chain, interacting with the hydrocarbon part of the membrane, it can stay there for longer time, right? So that stay period you can increase. How can you utilize it for intracellular interactions? So nowadays, a special technology has turned up for the nanomedicine that is called nano cases of DNA, right? So DNA origami. So DNA based nanotechnology. Now, what happens? You have a particular sequence on that nanosystem. That same nanosystem can be created on these nanoparticles because they have autofluorescence, and their fluorescence you can tune that from the same batch of particles. One particular population is giving green fluorescence, another is red fluorescence. You tag them with different sequences when they are inside the cell, and if they are interacting, there will be fluorescence quenching, and you can follow it, you can quantify it, that how much interaction between those two long chain nucleotides is happening inside the cell. You can, another concern which is very genuine concern, that what happens, whether it is safe or not, what happens, what is the fate of these particles. So we started investigating that what happens to these carbon materials where they are incubated with biofluids, when there are some particular enzymes available, and we found that they are completely degradable. They degrade inside the body and they degrade in soil because the peroxidase enzymes, they can chew them off. And we did follow the pattern of Raman spectrum to find out what is happening to the integrity of the carbon structure. We found that it was completely chewed off within 7 days in biological system or cellular system, you can say, and in soil, it horse radish peroxidase, which completely cleaves it off in 24 days. So basically, it is a sustainable technology which doesn't leave its own carbon footprint, because nowadays it is one of the catchwords, your carbon footprint, right? So this carbon material doesn't leave its own carbon footprint after being used or after being discarded. Then how can you quantify that how much drug is being actually used in the cellular system? So most of the side effects which we get out of medication is because of the overdosing, because we do not know. And the process of delivering is also, it's so unpredictable that once you inject, by the time it reaches to that particular tissue, only 4 to 5% is being used. Rest 94% or close to 95% is being cleared off from the system. Now that how to avoid overdosing and finding out what is the amount of drug in that particular state exactly required to do that either protein activation or deactivation. So these, because these nanoparticles, they have their chemical signature. So there are some spectroscopic methods which can be used for screening those drugs in a particular cell type in laboratories, and then can be given to doctor, and then doctor can give suggestion based on that, that okay, doses should be increased or decreased. So there is, here we used hyperspectral imaging, and we could read the chemical signature of these nanoparticles and the drug which was loaded on it, and we could see the separation time, that how much time it takes to separate and what amount of drug actually got separated. And based on that, we could decide our doses, and we did the animal experiment, and which was more successful rather than when we were doing without estimation. Then, as I said, it can be used for 2-photon bioimaging. Just the surface passivation of polylysine and the carbon materials are having very long half-life time when you are exposing it under a microscope for doing 2-photon. So that was really nice. Then it can be also used as, not directly as a therapy, but as a supplement. Now what happens, one of the biggest problem is the drug resistance, and that drug resistance is not only problem of the eukaryotic cells, even prokaryotic cells. So antimicrobial resistance is one of the domain. So what this material can do, it can carry different kind of salt content, different kind of ions, and make cells more susceptible. And then you can do the secondary therapy, whichever you have decided, and it will be more working. So here we try to do it for the GSH modulation, and we could deliver iron content, iron ions, entrapped on these nanoparticles using different kind of polyphenols, and we got the success in that, and we could improve the therapeutic potential of some of the conventional drugs. Then some gel material. Now we know that for the muscles, if there is a mechanical injury, if you leave it for a few weeks, by itself it will be cured. But right now, the way our country and all across the world, people are involved in sports and athletics, even one particular injury for one particular match can cost somebody a gold medal or somebody a very good big trophy. They don't want their player to wait for that long to recover. Now for that, there are some gel materials which can be used. Now, till now, what is the process, that you give some medicine, but the problem is that sometimes because of the ignorance, those drug molecules can create problem in the dope test. And without knowing, one of the metabolite of that drug can be caught in the doping scandal, and then that person is banned. Now that compels people to generate non-drug based material for regeneration of muscles. So we generated penylpan based or NEPAM based polymeric system gel, which could be used for enhancing the differentiation of myoblast to myotube. But the problem is that the strength of the material and the process speeding is not that much. So what we did, we used carbon material, and this carbon nanoparticle, when it went inside the gel, it not only improved the mechanical strength, but the adhesion and the differentiation process got speed. And within 72 hours, we could recover some muscle injuries, which generally doesn't get cured up to 10 days. Or so we did a DOMS model of animal, and there we showed all the biological parameters, including the biochemistries and histopathology, and we did extensive mechanical testing by using atomic force microscopy and other methods. Then these carbon materials can be included in biodegradable drug-eluting cardiac stents. where it can provide the strength, it can work as a reservoir for delivering the drug, and whenever there is degradation of the content, slowly it can release the carbon particle, and those carbon particles are further getting degraded and will be in the system for longer time. So it can be used there as well. So these are the ways we did it. Different kind of, I know that I'm a little late in that, but we did all kind of characterization. We got successful. We finally used it in ex vivo condition of the pig heart, and we could use those structures which we have created by 3D printing. These are some of the other technologies which are working, and these carbon nanomaterials are really handy in those cases as well. We are making medi patches, we are making medi caps. Those can be used for personalized medicine. That is another catchy word, but everything boils down to a very nice, sophisticated material which can do the job. We also generate biosensing platforms where we use this carbon material to coat on filter paper, and that paper converts to a biosensing platform. So that brings down the cost, and that also becomes like use and throw, and you can put multiple detecting molecules using antibodies or aptamers or peptide sequences to recognize a particular biomolecule which could be present in any of these biofluids, including urine, sweat, saliva, or tear, or blood, and then you can find out. So these are some of the examples and patented technologies which we produced. So finally, to conclusion and way forward. So you need a therapeutic regime, you need a method, but you also need a material composite which can do job for you. And thank you very much for your kind attention. And I'm open for questions and comments and suggestions, please.